Okay, good morning, everybody. I think we left off right here with Punnett squares. Hopefully you guys feel comfortable with this stuff. Are there any issues or concerns? Feel good about Punnett? Okay, so quickly, I do think we talked about this, but I'm going to start off right here just because it seems like a good point to kind of review the littlest bit. We're going to look initially with this one at a monohybrid cross. So just one thing, in this case, the P color, right? There are others, and we start thinking about maybe the P color and the P shape or other things. And we could call them dihybrid or multiple ones, tri or whatever, all right? We typically try to simplify things and not do like all 7 of them at the same time. Not that you couldn't do that, right? If you were trying to do a Punnett square with all 7 different characters that Mendel looked at, you would have many, many boxes. It would be humongous, right? So initially with this monohybrid cross, we've got true breeding plants that we start with. So always yellows and always greens, the gametes that they can produce. We're only showing you one, but they are homozygous. All right. So there are 2 of the same gametes or same alleles, copies of that gene. All right. So big Y's, little y's, and in this F1, the offspring of this cross, everybody is heterozygous. 1 dominant or capital letter and 1 recessive, right? And the dominant masks the recessive condition. That happens when we think about Mendelian inheritance, when we think about dominant and recessive characteristics, right? Not everything follows this pattern, and we'll hit that in the next couple of slides, all right? Most stuff we think about following like this, where the dominant allele overshadows or masks that recessive allele, all right? And I oftentimes think good copy, bad copy, but that's not 100% true. So in this case, the gametes that we can generate from this progeny would be 1 of each, right? It could be a big Y or a little y. And if we allow these guys to self-pollinate, like we talked about in the previous lecture, we've got the opportunity to see some of those green guys that were up above that were masked in this F1 generation, they show back up, right? So with that, as we do the Punnett square, we bring the gametes from the F1 onto the side or onto the top. Each is heterozygous, so yellow and green, as we see, right? And with that, we drag them across and down and fill in the letters. For ease of kind of record keeping, it is wise to put the dominant allele in the front, regardless of where you may be finding it. But if you draw them specifically going across, you would see the recessive first and then the dominant, but it, again, dominant masks, all right? So with that, we could see the phenotype, the outward appearance, yellow peas and a green pea, 3-to-1 ratio, right? And drawing the letters or those alleles into the respective boxes, we can also see the genotypes, the combination of those alleles. So one of them that's homozygous dominant, one of them homozygous recessive, and 2 of them are heterozygotes, right? And that's what the bottom is trying to show here. I can get my mouse to work. The phenotypes, yellow and green, outward appearance, and then their respective genotypes, what you get. And with that recessive character, we know those alleles have to be 2 lowercase letters because that's the only way you can get there. But the other genotype, we're not sure, looking at those yellow peas, what their actual genotype is. It could be homozygous, or it could be heterozygous. And in both instances, we're yellow, right? So just as we counted them up there, there's 3 ways to be yellow and 1 way to be green, all right? 3-to-1 ratio. And I mentioned this, Mendel did these experiments and collected this data with thousands of pea plants, not just 1 pea plant, all right? And in turn, taking all of that data gives him the ability to come up with these ratios. It's not exact 3-to-1, but it's pretty stinking close, all right? So lots and lots of data, massive amounts of peas, all right? So in this case, we're looking at a dihybrid cross. So pea color, yellow and green, and we're also looking at the shape, round and wrinkled, right? And as it turns out, we already know that the yellow is dominant, but as it turns out, the round is also dominant to the wrinkled, right? And we can see it because of the way we've got it expressed up there, all right? So in a monohybrid cross, 4 window panes that you're building to fill in the specifics. And in a dihybrid cross, we've got considerably more, all right? So 16 windows for a dihybrid cross, and we keep compounding it the more we want to do. We're not going to go anywhere farther than this, all right? And test-wise, I think it's really just monohybrid cross. However, let me back it up 1 second. This is technically important right here as we see these specific alleles or these chromosomes that we have in this F1 generation, all right? And what we get to bring down here, you want to understand what's happening there, and I'll walk you through it here in 1 second. So with our first, the top of the figure, with that P generation, we've got 2 true breeding plants, yellow and round and green and wrinkled. And they've been self-pollinated a handful of times, and we always get the same stuff, all right? So now we cross these 2, and what we get is a hybrid, a heterozygote, and it is one that has the mixture, all right? But it still looks yellow and round, all right? So if we look at the alleles, there's a capital and lowercase y and a capital and lowercase r. It's heterozygous, all right? So those respective alleles that we can have, all right? If we start with the capital Y, all right, and I bring it down, 1 big Y, if you can't have 2 of the same letters together, not in your sex cells, all right? They have to be different, all right? But we've got 4 potentials, all right? So big Y to big R, that's my first one. Little y to big R is my second one. Big Y to little r is the third. And last are the little guys, little y, little r, all right? 4 different potentials, all right? That's why we've got 16 squares, all right? So 4 possible combinations, same as we go down the other side. It's the same, we're self-pollinating, it's the same thing, right? And similarly, for kind of easy record keeping, I try to keep the same way I wrote the stuff on the top to be the same way going down the side, right? That's a little bit easier as you're moving through there. Then you simply bring in the letters, right? Couple capital Y's, couple capital R's, so on and so forth. We move through there, all right? Hopefully that makes sense and seems pretty logical to move through there, all right? When the exam rolls around, think about some of these things, all right? And like I said, really more monohybrid cross, but you could draw yourself out of Punnett Square to kind of think about some of these potentials. Like, what are the chances of being heterozygous in a monohybrid cross? Hopefully, you know that's 50%, but if you don't, you could draw your quick box and see there's two ways to have a capital and lowercase letter, two squares, right? So enough of that. As we look at this cross, there's a lot of yellow and green going on, all right? We're looking at it all together, right? But we also can break it down just a little bit, right? And we can count up all of these things, and we could say yellows to greens, which is, as we reduce it down, is a 3-to-1 ratio. We could look at rounds to wrinkles, and it's the same, 3-to-1 ratio, right? That we get if we're looking at the single characters, right? When we look at the dihybrid cross and we start counting everything up, we get a different ratio, not just this 3-to-1 ratio, because we're looking at multiple characters, right? So the phenotype for those that are round and yellow, there are 9 of them, and you can count them up, all right? 9 round and yellows, there are 3 round and greens, all right? 3 of them that are wrinkled and yellow, they're right here, right? And only 1 of them that's wrinkled and green, like what we started with up above, right? It's a similar situation, right? With this kind of 3-to-1 situation, all right? In order to have the wrinkled and the green characteristic, that the odds of that are very small, all right? As we look at all of these potentials. So multiple things can be evaluated, multiple traits. And the kicker with this, and fortunately, really, for Mendel, is the fact that each of these specific things that he tries to evaluate happens to be on a different chromosome. So evaluating the round and the yellow or the whatever, separate chromosomes. And as they segregate, as we separate these things apart, that gives us the opportunity to evaluate this stuff. If we had the green and the wrinkled on the same chromosome, well, then you would expect it to be similar probabilities or similar amounts as it's passed on, right? So in that case, we wouldn't be seeing this 9-to-3 to 3-to-1. It would be a simple 3-to-1 ratio because they would be on the same chromosomes, right? That's not what we see when they're on separate chromosomes, right? It gives us this variation, right? More detail. And truly, I think, pretty fortunate for Mendel that it was that way. Could he have figured out something else going on? I'm pretty stinking sure, all right? So we will get to some of that here in just one quick second, all right? So this goes to kind of what I was talking about as we see the potentials with these chromosomes, and on a separate chromosome, that specific gene for that specific character or trait, right? So up here with our first, so it's 2 different potentials. Our first chromosome arrangement, we can see in metaphase 1, we've paired up our homologous pairs, blues on one side and reds on the other. We separate through meiosis, the second round of meiosis. We see metaphase 2, separating only blues or only reds, and the gametes, that's all you get, all right? Blue or red, all right? Down below is some of this variation, and this is the, the independent assortment as we see this, all right? Is Mendel's second law, right? As we see these chromosomes with multiple potentials, right? For how they segregate, right? And with that said, that gives us opportunities to see things like this, right? As we move through there, as they segregate or separate into their respective sex cells. So here, as we look at this, to kind of put it into perspective, we could think about round and yellow and wrinkled and green. And then you get over here and we could be round and green, right? For this one, right? Or the other way around, whatever, right? There's too many of those for me to start spouting all of that off because then I get all confused. Okay, so that's about it when we start thinking just this Mendelian inheritance, right? Dominant, recessive. We still have same idea with chromosomes. We've got different alleles. You've got 2 copies of these respective characters, one from each parent, right? The ways that they are expressed, like I said a second ago, oftentimes it fits what we just talked about, Mendelian inheritance, classical Mendelian inheritance, right? But sometimes it doesn't quite fit, right? And this kind of goes back to those initial thoughts when you reproduce and you shuffle these genes and now you've got something completely different. And once that happens, it's a done deal, right? That's not the case, right? But that's what the thought was at the time. And Mendel set out some studies to try to show that wasn't the case, right? Here is an example of that, snapdragons. And these are flowers that there are all sorts of varieties of colors, right? Along the building out here, there was a whole bunch of them, right? Maybe there still is some of them out there. So in this case, the pink flowers are heterozygous, right? And it is a mechanism that we call incomplete dominance. In this case, it's the cross between a red flower and a white flower, right? With the pea plants, a purple flower and a white flower gave us a purple flower because purple was dominant. But in this case, a red flower and a white flower gives us a pink flower, right? Incomplete dominance. And so basically, as we see this with the incomplete dominance, it's like, well, they're both being expressed. You add some white to some red, you get pink, right? You add, you know, a blue flower and a white flower, that's not as simple to see. It's light blue, it's masked, it's downgraded or dampened the littlest bit, if you will, all right? Incomplete dominance is the partial expression of each of those alleles, all right? Where the white flower is still expressed and it masks some of those characteristics, in this case, making pink flowers, right? But it's not a shuffling, a mixture, and what you see is what you always get, and every pink snapdragon is going to make more pink snapdragons. That's not what happens, right? So with this, the pink, and you could, oh my gosh, that's a big honk of chalk. That's like sidewalk chalk. It's like my kids have been in here when they were young and wanted to do sidewalk chalking. So as we look at this with our Punnett square, our pink snapdragon, all right, we will call the heterozygous condition a big R, little r, right? And that gives us pink for our snapdragon, right? Really, yeah, I think that's what no, that's not what I want. We want this, an R and a W is pink, right? So big R is red, W was white. It took me a second to recall what was going on. So that's what happens if we cross our big R, big W, we get RWs, and every square would be RW. A cross between a red and a white snapdragon. All of those seeds, as you grow them up, gives you pink snapdragons, right? Now, we can take some pink snapdragons and it's one of each, right? So we can have an R and a W, and an R and a W. And when we do this Punnett square, we wind up with one of them that's red, one of them that's white, and two snapdragons that are pink, right? It's similar to what we've just thought about with Mendelian genetics, right? It's the littlest bit different, right? Instead of one capital and lowercase, we've got two different alleles that we're looking at, and they both participate, all right? So the white downgrading or reducing the output of the red, making them look pink, right? So I'll write the phenotype on these guys. And white. Okay. That's incomplete dominance, right? There's other stuff too to lend to some confusion. So there are things that are codominant. So they're not down regulating or reducing stuff. They are both expressed equally, right? One great example of that is with our blood typing system, right? There's a couple of things that we can see here, all right? There are some alleles that are dominant to other alleles, and there are a couple that are equally expressed, right? So a few things, all right? In this case, the I here, what is that signifying? I should remember this off the top of my head, but I'm not right now. It doesn't matter, right? You've got a handful of different potentials for your blood type, all right? 4 different types, A, B, AB, or O. Maybe you know what you are, maybe you don't, right? But dependent upon that, there's a couple of ways that you can be a specific blood type, all right? To be type O, there's only one way, all right? So as we look at this situation, we're looking at a codominant cross. We're looking at a cross between 2 AB people, all right? And as we move through here, you will see down below, there's no chance that O is in this mix, all right? It's not up there, all right? We'll show you that in 1 second. So one person AB on the top, the other person AB on the side. And as we do this cross, we've got one of them that has 2 A alleles. They are type A. We've got another person with 2 B alleles. They are type B, right? And this person right here with one of each, this is the codominance, and they are type AB. Both of those alleles are equally expressed, right? There are definitely some differences with this, where we're more than just type A or type B or whatever. We think about other things like positive and negative. The RH factor is another thing, and we'll hit that here in just a second. So you maybe know a little more about what your blood type is based off of some of that, all right? So from this cross, 2 AB individuals, right? You can see some of the potentials, 50% chance of having AB offspring and 25 of the other possibilities, right? So this is only with these 3, right? And we can see, you know, technically we would think, you know, kind of heterozygous, one of each copy, right? And so 2 ways to be that, right? So with the other, all right, and this, so the little i's, it's A and B, they are dominant to O, and I think that's why it's the capital I. And then the little i's, 2 little i's, and that's what you've got, all right? So as we look at this, there are a handful of different potentials, all right? You've only got 2 alleles, all right? But we've got multiple opportunities up here, just depends on who you are, all right? We call this multiple allelism. So there's more than two alleles in a population, right? There's A alleles, B alleles, and O alleles, or the little i's, right? Dependent upon what your combination is, gives you your respective blood type, right? We're still not talking about positive or negative, right? So up here, we can see, looking at the dominant stuff, this is what we've already shown in the previous slide. And then we're slapping in the incomplete, or the recessive, sorry, not incomplete, but the recessive character that is a potential, right? So we saw this. There is another possibility for you to be big A, I big A, superscripted, and a little I, right? So one allele for O, one allele for A, but A is dominant to O, so it masks it. You are type A, right? Same idea with B, a little I with the B, you're type B, right? Same way going over here, it's just different partner that's contributing that. So now you're type A or you're type B, right? If you are type O, you know what your genotype is. Gotta be this, right? It's like the, that little wrinkled green pea as you do that, dihybrid cross. That's the only way you could be that, right? Is by this, right? Everybody else, there's multiple ways that they could be B or A, technically AB as well, as we see here, BA, AB, right? There's multiple ways to be that, right? So maybe we think about stuff like blood tests, paternity tests. This is sort of accurate. It's not 100%. Just because you have the same blood type doesn't mean that that person is your father, right? It could, and there's other genetic stuff like DNA screening that lends to more details. We really think more with the blood tests for ruling out, right? Not for saying, yes, this truly is your father, right? So it depends, right? So you may know your blood type. You may know your parents' blood type, right? And with that, you can think about some of those potentials, right? If you happen to be type O and one of your parents is type AB, you might want to ask some questions because there's no chance that that person could be your parent, right? What the hell, right? You don't want to learn that in college biology, right? But sometimes that's when students figure some stuff out. I've had a handful of people that are like, oh, I had a friend in anthropology class because they talked about this in anthropology as well. They're like, that's when they realized it. I was teaching this class up in Idaho Falls at the community college. And I had a lady that told me same idea. She wasn't, it was, many years later that she realized that her dad wasn't her dad, right? And she confronted him. He's like, well, I've been your dad your whole life, right? What's up, right? And there was no, she was all fine with that. But some people, it destroys them, right? Definitely some potential issues with that, right? So this multiple allelisms, there's more than one way to get this, and there's also codominance and regular dominant recessive stuff with the AO-BO situation, right? So type A people or type B people, if we are in this category right here, they do harbor an O allele, this little lowercase I. And an A person and a B person could technically create an O person, right? If they have those respective alleles, right? What about an O person with another O person? What would their children be? What do you think? Oh, spot. Oh, yeah, you knew it. You could only be O, right? That's the only way you could be that way, all right? So multiple ways. AB with AB, you're only going to be AB, right? So the RH factor, I realize it's not on there. We'll talk about it real quickly. This is important when we think about, it's really important when we think about women, right? And ladies that are hoping to have children. That's where the positive negative is an issue. Way back in the day, you know, love is in the air. Two people decide they want to get married. In order to get married, they made you take a blood test. You would go in, they would test your blood and basically see if you're compatible or not. Really, they're looking at the positive and the negative, right? And if the mother is, if the woman, she's not a mother yet, right? If the woman is negative and the man is positive, there were potential issues, right? And oftentimes those people were told, you can't, we're not going to give you a license because you're incompatible. And I think about that, if love is in the air and you decide you actually want to get married, is a blood test going to prevent you from having that happen? Maybe, maybe not. Is a blood test going to prevent you from having intercourse? Probably not, right? And you know, today we don't really give a crap about whether you're married or not, right? You can reproduce whenever the hell you want, right? And do you get a blood type before love is in the air? I bet not, right? Doesn't happen, right? So there are issues, or maybe I should say there were issues. There still could be issues, right? But we recognize that there are these specific problems, and we also recognize we've got abilities, we've got drugs that can prevent the production of antibodies that will target these issues, right? So if a negative woman is pregnant with a child that happens to be positive, on the first pregnancy, it's not really that big of a deal because this is like the first inoculation. This is the first shot, if you will, and it takes 2 times really to mount a big response. So the first child stimulates an immune response within the mom as the positive blood is detected by her system and she produces antibodies against it. The next pregnancy, if it happens to be positive, she's already got antibodies produced and she goes into overdrive to try to fight this foreign invader. And if it doesn't die, there are serious complications as a result of it, right? It cause what we call hemolytic disease of the newborn. You want to see some nasty pictures, Google image that. So we know there are problems, all right? There is a drug called Rogam that blocks the production of those antibodies. And so if this happens, you take that shot, you take it a little bit before the due date, and then right as you go into labor, you get another dose of it, and it blocks that production, right? Now, a negative woman and a positive man can have children, and there's really not that big of a deal, all right? Because we can block that production, all right? One quick little story that I thought was sort of interesting, and it made me kind of stop for a second. I'm O negative. My wife is O negative, right? Both negatives, there's no way you can make positive, right? 2 negatives make negative, right? Because the RH is simple Mendelian genetics. If you're negative, you have, well, you basically lack those 2 copies, right? If you're positive, you could have 1 of each, right? And a positive person could create a negative or a positive, right? But 2 negatives only get negative, right? So with our first child, we're at the doctor, midwife. We're in there for one of these appointments, and she looks at my wife and says, "Oh, we need to schedule your appointment to get some Rogam." And I looked at her and I said, "Oh, she doesn't need that." And she looks back at me like, "Who the hell are you? How do you know what she needs?" Right? And I'm like, "Well, I'm O negative," right? And she's like, "Oh, are you sure?" Yes, I am. And I pulled out my Red Cross blood donor card and I showed it to her. She took it and made a photocopy, came back. She looks at me, she looks at my wife, and this is what I'm like, who is this lady, right? She looks at her and she's like, "Are you sure he's the father?" And I'm like, "What?" So I look him up because I'm like, maybe this is a question that, what's the answer going to be? guess, right? I'm like, that seems kind of odd, right? But both of my boys, O negative. Does that mean I'm their dad? Not 100%, right? Just because we got the same blood type, but I'm pretty sticking sure I'm their father, right? So if they were anything else, well, that'd be a different story, right? So kind of odd. She did not get RhoGAM. She didn't need it, right? Secretly, she calls the doctor. She's like, actually, let's schedule that shot, right? That's not what happened. All right. So here we're looking, these Drosophila is a fruit fly, right? And, you know, there are all sorts of fruit flies around right now as, you know, stuff like tomatoes coming into the house. We see little fruit flies, dang you buggers. So there's all sorts. Drosophila is a model organism. It's something in biology that's technically pretty easy to grow, to manipulate. The genome has been sequenced. It's got just a handful of chromosomes. It's easy to understand, right? There's been all sorts of research with fruit flies. There's been research when we think about like looking at evolution in a laboratory situation, right? Something that we can control and manipulate, right? Really pretty impressive, all right? So in this case, we've got eye color that we're looking at, red versus white, right? So the red eye color is wild. Wild type is the natural, the normal situation, right? And it's dominant to the white eye color, the mutated form, if you will, right? So it's on X chromosome. And with that, we think about the potentials for thinking sex-linked characteristics. So here, red versus white, and in the males, only one X chromosome. It's all blurred out here, but it's a capital W, right, for the white eye. And the female is one of each, big W, little w, whatever. So it just depends. And we've got a cross up above with a male that has red eyes, the capital, and the female with white eyes. I don't know why we've got them that way. Whatever. That's fine. So this showing a sex-linked trait and the potential with this is definitely more common when we look at males versus females because you only get one X. You only get one of those copies. And in a female with two, there is a possibility of being heterozygous, and that's definitely more likely than being homozygous recessive, right? 50% odds of being heterozygous and 25% chance of being homozygous recessive, right? So there are numbers of ideas when we think about some of these sex-linked situations, the X chromosome, color blindness in humans as a possibility. And we discussed a little bit of that last week. So we talked about crossing over before. When we think about the generation of the gametes, the sex cells, as this is occurring in the first round of meiosis, as the homologous pairs, or as the homologous chromosomes pair up in metaphase 1 of meiosis, they don't all do it, but there is a potential as they're held close together that some of these chromosomes can intermingle, if you will. And this crossing over event provides differences to these chromosomes that they didn't have before. Lots of blue, little bit of red, or vice versa, all right? The recombinants are the ones in the middle. The ones on the outside don't recombine. They're the non-recombinants. They're the same as we had started with, all right? So this provides variation, right? The random assortment, the independent assortment with that, and the crossing over all gives us potentials to have alterations in these chromosomes within the gametes, right? That aren't. the same as what you see in your own normal body cells, your somatic cells. Different. So this, and I think we might be toward the end. Well, we are. This is pretty interesting, and there are other examples. I'll tell you about some dogs here in 1 second. But epistasis is a situation where one gene acts like the switch. And if you have that gene, we switch the switch on. And if you don't, it stays off, right? And what that switch can control, there's all sorts of different things. But in this case, we think about coat color, all right? So there's all sorts of different colored mice up here, right? As we look at these guys, albinos, black ones, and agouti, these are the types, so kind of brownish, right? With epistasis, this gene in this case, C, masks the expression of another, all right? And the other will be A for the coat color, all right? And A over here for agouti, which is the dominant, right? As you see, the bulk of them are that color, right? So in this case, right, we've got two that are heterozygous, big A, big C, little c. The big C, doesn't matter if it's 1 or 2 copies, the big C flips the switch on, now we express the color. The big A, in this case, says, this is your color, right? This brownish color, right? So as we draw the respective alleles that we can have doing this dihybrid cross is really what we're looking at, all right? 2 different things, A's and C's together, all right? As we do this, we can see homozygous dominant, 2 capital A's, 2 capital C's, and a lot of heterozygous, big A, little a, whatever, all right? 9 ways to get this, all right? There are 3 ways. The 2 lowercase a's gives us the black mouse, right? So big A, brown, little a, black. Little c's, all of these white mice, the albinos, all of these, doesn't matter what the A says, the little c's, 2 of them are there. So it doesn't matter if you're big A or little a, there is no, the switch is shut off. The little c's are there saying, I don't care what color you want to be, you're albino, right? So that is epistasis. The C allele says express it or don't, coat color or not. Big C for coat color, little c, no coat color, all right? And you get a bunch of little albino mice, lab mice. And oftentimes, there's a lot of homozygous stuff when we think about laboratory animals, lots of albinos where you know specific genetics and you know details about them, right? Kind of interesting, right? There's all sorts of variation when we think about dogs, right? There are little teeny dogs and great big huge dogs and all sorts of variation with coat color, all right? One example that fits this would be Labrador retrievers. And you can see some black labs, and you can see some yellow labs, and you can see some chocolate labs, all right? And with that, it's the pretty much exact same thing that we're looking at here, all right? The yellow lab is this, right? It doesn't matter if we're talking coat color or not. Black, I believe, is dominant to the chocolate, but I could be wrong with that. I'd like to think that it matches this exactly, but I think it's maybe a little bit different. I think chocolate labs are more in this character, right? But it's a similar idea, right? You express that color, your big C is there to express it, and either way, right? So the black lab or the chocolate lab, all right? And to be a yellow lab, it's little c's, regardless of the specific coat allele, the color that you would be exhibiting. Does that make sense? Clear as mud. Do you guys have any questions or concerns? Feel good? You feel confident? You ready to take a test? Okay, that's all I've got for you. The test will open. We do not have class on Thursday. The test opens, I think at 10, maybe 9. I can't remember what I made it open at. I think it's 10. When does this class start? 9. 9:30 it starts. I think the test opens at 9. I knew it was a little bit before. Okay. Issues? Anybody from home? All good? Okay. Have a great weekend, guys.